This is a demo of Salmon RNA-seq workflow. Let's start by loading the workflow here, and under our Git pod, we have the workflows directory, and it'll be called salmon_demo. So let's choose that directory, and once you choose that, it'll show this as the workflow. So a bit of a background of this workflow. Pretty much, this is kind of inspired off of the official Salmon tutorial they have under the website. So if we click on, go to the website here, and they have a get started page. And in their get started page, they go through how to, at least in their tutorial, how to set up Salmon and get some quantifications and gene expression out of it. So kind of like just a bit of like parallels of what we have with what they have. First steps they have is just to kind of how to install Salmon. I think the first way of recommending is to use Bioconda. Then the second way is to use their binaries off their GitHub for Salmon. And the third way they said to do is to use their Docker image, which under their Docker Hub, they have an official image for Salmon, which you're going to pull from them and then use that in our workflow as a widget in this case. So going through the workflow itself, it runs in a left to right manner, starting with the start widget, which we specify all of our inputs to use in the workflow, especially with these downstream widgets. We gonna have to have these entries, specify these entries here, and then these entries and parameters be thrown to these other widgets as inputs for those. So we take these entries here, and they become outputs for, or become inputs for the other widgets. In this case, we start off with the work directory, which is going to be where we store all the files that we either downloaded or are generated from these other widgets. So we could have this here, salmon demo work. If you want to change it, we can. Just type in what kind of work directory name you want. So if you want a second one, you could type in 2. But for our purposes, we'll just keep things as default. The second entry here, SalmonIndex directory, which will be going to be the directory that we specify when we build the SalmonIndex in this widget, which I'll explain later once we get to that widget. These other entries, sample base names, sample download links. So this is based on what the Salmon tutorial they have, what they used as samples and are their species that they want to use for their tutorial. If we go down here, quantifying samples or obtaining sequencing data here, they specify where they got a project to use for their tutorial. So if we click on this link for this project, you'll be brought to this ENA browser and to this project where they use Arabidopsis thaliana, which Googling it is going to be this plant as their species to use. They have a bunch of samples for that species, and associated with that, they have IDs to them plus FASTQ files. As these FASTQ files are paired in, so each sample has 2 FASTQ files associated with them. So you'll see a pattern here. If you see the DRR and then a bunch of numbers here, it's associated here with the FASTQ file names. So same ID, run accession ID they have, and then underscore either 1 or 2, and then FASTQ.gz. So that's kind of a pattern of how they name it if you want to download it. So I'll explain how to pull, copy and paste these entries, or these names and download links into our start widget and exercise that'll be coming after we first run this workflow. As I pulled up a name here, a base name for a sample off the website, and then plus the 2 FASTQ files that are linked to that sample. And you see the links here, they're currently dashed because it's not been executed yet. The start widget has not been executed, so the outputs from start will be inputs for these other downstream widgets. And once start widget gets executed, you'll see these lines will become solid. They're basically just drawing inputs that we specify in the entries, and it'll be thrown into these other widgets as inputs. So let's say, for example, work directory. So workdir is going to be our work directory set up here, and it'll be thrown into this entry called directory, which in this case is a download directory. So that's kind of like why it's kind of grayed out. So it'll automatically be thrown from the start widget into the next widget that comes after it. So this widget is at the top here. This is to download the FASTQ files, which we specified in these two links. It'll be downloading from there into the work directory that we specified in the start widget. And then this widget for download widget down here is to download the FASTA file, which is our transcriptome, which we're going to use eventually to build an index, because Salmon needs an index in order to quantify. So that's the third step after we download the FASTA file, is to have this SalmonIndex to build the index to use using the transcriptome, which is going to be the same, using it under the same species. Going back to their tutorial is pretty much this link, and then just download this .fa, which is the FASTA file. So that's where I'm getting it from, which is under the step obtaining a transcriptome, building an index. So we convert this command to how to run it in this, under this widget, which we have more parameters to set up, but we'll just be only specifying the number of threads to run it to make it faster. We don't have to change any of these other entries or parameters. So we'll just keep things as a default, keep things simple. After we built the index and after the FASTQ files are downloaded, we'll end up in the quantification step, SalmonQuant, where we have the SalmonIndex directory, which we specified in the start widget, and then output quantification directories, which will be automatically be generated from the start widget, as there is kind of a Python script that runs in the background of start to kind of, it knows from, based on the base name, it can use the base name and tell it, okay, so it'll be, the output will be thrown into a directory under using its base name as kind of the base directory name. In same case here, the two FASTQ files are specified based on what the base name was. So it's like drr something 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 and then _1 dot FASTQ, and then it'll be same thing for this one, _2 and then dot FASTQ kind of thing. Same number of threads for speed things up. And then the last step is to quantify or show the quantification file that comes out of the quantification step, which will be thrown in. In this case, we used the base name here, _quant, which is our directory, and then quant.sf is the file that we show for gene expression for all the genes that are listed for that sample that we specified at the beginning. So that's pretty much a rundown of what the workflow entails. And if we're going to start it, I'll start it right now. Going to show what's going on here. Everything is run using Docker containers. It looks for the image first, but we have to download it if it's not there under Docker Hub, which we have available. And then once that's finished, you'll see that the dashed lines that were originally dashed, but now solid because it finished running, and all the parameters that we specify here get thrown into these downstream widgets as inputs. So you see FASTA files download quickly, which we see SalmonIndex running. Currently FASTQ, download FASTQ step is still being completed. So once this is finished, then you'll see this line be solidified. But at the moment, you'll see, because SalmonQuant is waiting for these two widgets to finish, all these dashed lines need to be solidified in order for this widget to be executed next. So we have just got to wait until we find these two widgets to be completed in order for SalmonQuant to be executed next. So just opening up one of the consoles here, you'll see whatever the tool is thrown in. Oh, it's finished here. So looks like these two widgets are finished. So you'll see SalmonQuant is now running next. And then what you see here is what SalmonQuant, there's kind of steps that is being executed currently. What's thrown in out from the tool is being thrown into the console. as it's running. It might take some time as it needs to do some the quantification, do some mapping and quantification of that. So it'll take some time for that sample in order to get some gene expression out of that. So I'll just let it run and wait for it to be completed. And hopefully I should take like a minute or so to complete. Okay, so it looks like that was faster than I expected. So now the next step is to show the file, the quant.sf file we get from the output from Salmon. Currently, I think, yeah, so it looks like the Gnumeric Docker image is not found, so I'll be pulling it from it directly. So it just takes some time to pull that image and then build a container. So here we go. So this is the output from Salmon for that one sample that we specified at the beginning. You'll see the name of the genes, and then looks like the links of each gene. And then I think the one we want TPM, which is normalized counts of that. So that's pretty much how the workflow runs from start to finish. And in the next step, I'll explain how to change, if you want to change some of the entries at the beginning. If you want to look at another sample, we'll go through that, which will be kind of like how to use copy and paste tools, and then see if we can get some outputs for the second version or another sample to use. So be lookout for the next video to go through how to do that.